Hello students and welcome to my pre-AP Biology Chemistry Review. This video is meant to help you out if you haven't had chemistry in a while, and it's helped to get you up to snuff so that you're ready to do Unit 1 in AP Biology. It is not meant to be the sole instructional video for Unit 1. That will come later on. All right, so like all my videos, this is not for profit. This is solely meant to help students. And so that is the license here. Images come from Khan Academy and OpenStax, which graciously allow people to use them. And you can access them at khanacademy.org and openstax.org. So let's dive in. This comes from the AP Biology student website. And it says that, AP Biology is supposed to be a college level course, and you're supposed to have had biology and chemistry beforehand. Here at TASM, we've made the decision that you can take AP Biology at the same time as you're taking high school chemistry. And I'm okay with it because I think that we should encourage as many students to take AP Biology as they want to. And if you're willing to put in the hard work to understand the chemistry needed for AP Biology, then kudos to you. And so this is kind of meant to help those students. If you've had chemistry or if you've had AP chemistry, you probably don't need to be watching this, and you could just do a quick review. This is what Unit 1 Chemistry of Life looks like in AP Biology. It's going to go over water, hydrogen bonding, elements of life, and then really lipids, carbohydrates, proteins, and nucleic acids, and dive into those. But to really understand what's going on here, you need some basics. Like you need to kind of understand, why is water a polar molecule? What makes that happen? Why are these fatty acid tails nonpolar? What goes on with protein folding? What is a hydrogen bond? What are van der Waals interactions? And so that's what this video is meant to be. It's meant to be the basics to help you get ready for this unit. This is from my observations, having taught AP Biology for the past several years, that there are no direct chemistry questions on a test. That's on an AP chemistry test. And they've even removed things like Gibbs free energy and enthalpy questions. But giving you an SAT word, it behooves you to know a bit about chemistry as you think about how systems process energy and maintain homeostasis. I'll give you two examples here. Knowing a little bit of chemistry gives you some idea about why water is a polar molecule, and then how those polar molecules, when they interact together, give water unique emergent properties that makes water crucial for life. And then there's stuff like the mitochondria, right? Everybody loves to say the mitochondria is the powerhouse of the cell. Well, it actually involves a lot of biochemistry involving these hydrogen protons, and then differences in pH and concentration gradients and electron carriers, etc. All right, so when I put this together, I kind of went through Khan Academy and looked at, well, what do they say is the chemistry of life needed in a biology course? And you'll see we didn't get to a lot of this in my 9th grade biology class because we were doing the next generation science standards, and it dealt with more of a holistic look at micro and macro parts of biology. So you need to know about elements, atoms, how valence electrons work, how do they form compounds, and bonds and reactions. And so let's jump in. So pause the video. Can you answer what is an element? Okay, hopefully you got something along the lines of carbon, oxygen, nitrogen. It's probably easier to give examples of them. But an element is something that can't be broken down into anything else more simple. Kind of like a cell is the basic unit of life, an element is the basic unit for this chemistry. And so here is the periodic table of elements that arranges them according to properties and is very useful as we look at different elements. What is an atom then? How are atoms made? So an atom is the smallest unit of matter of that element. So you would have gold atoms or silver atoms or carbon atoms or oxygen atoms. So a gold coin is made of mostly gold atoms put together, and then a few trace other elements. And how are all these things made? You've probably heard of this before, right? That they're made as stars. And so here's Carl Sagan saying it. And I encourage you to check out Cosmos with Neil deGrasse Tyson. There's an excellent evolution episode in there. But everything that's an element comes from collapsing stars. So you are made of stars, students. And you are a star in all of our eyes. And so these stars are running on nuclear fusion, putting together hydrogen to make helium. And then as they start to run out of energy, they do other fusion reactions, which make heavier elements. And then when they blow up, they can make elements that are heavier than iron in supernovas. So we all have a bit of stars inside of us. So we're all made of stars. Okay, the parts of an atom. And so hopefully you remember this from middle school or from whenever last time you had a little review of chemistry was. There's going to be protons and neutrons on the inside of an atom and electrons on the outside. And these protons and neutrons have mass, and the electrons are very light. And so the electrons circle around. You can think about them like orbits. And so you might have learned, depending on where you studied, etc., you might think of the orbits as just circles, or you might think of them as orbitals, like dumbbells, etc. And we'll jump into that in just a little bit. Okay, what is a compound? Well, a compound is two or more elements combined together, right? So here, combining together hydrogen atoms and oxygen atoms, you can make water. And so this is different than just hydrogen or oxygen by themselves, and it's going to have different properties. You combine together carbon, hydrogen, and oxygen, you can get a carbohydrate, like glucose. You could also make a lipid, like a fat. You combine together sodium and chlorine, and you can get table salt. And so that's a couple examples of a compound. All right, so here's an activity that we do in class and that you'll enjoy in AP Biology. When we get to it, you get to try and sort these cards in order from largest to smallest, and argue why. And you can't use the internet. You can't look up what UM and PM and NM stand for. And you'll see just how small an atom is. And that's from the Learn Genetics website, so you can check it out. I'll link it below. All right, so everything's made of matter. Matter's made of atoms, and elements are made of one type of atom. And so here are just some models or some ways of showing what different atoms look like. And this should hopefully look familiar. So here would be hydrogen. Hydrogen just has one proton and one electron. And here's oxygen. So oxygen has 8 protons, 8 neutrons, and 8 electrons. So you may be wondering, how do I figure this out? Where do I figure out protons and neutrons, and how do I know which orbit these electrons go in? And is it really like this? Is it like this in the real world? I've heard of things like Schrodinger's cat and stuff like that. And so let's dive in. What you need to do with that is you need to be able to check out a periodic table. So there's one in your textbook and linked in Google Classroom. In biology, we'll mainly focus on the elements SPONCH. And so SPONCH stands for sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen. These are the elements of life. Other elements are also very important, and they serve as cofactors and enzymes, and they make things happen. But these are the ones that we will mainly look at in our chemical reactions. So let's look at one. Let's look at hydrogen. Here is hydrogen on the periodic table. It has an atomic number of 1, meaning that it has 1 proton. Its symbol is H, just the first letter of its name. And here is its relative atomic mass. It's going to encapsulate all the different isotopes, and it gives you the name of it. So let's look at oxygen, which was on my previous slide. And I said, how do you know it looks like this? How do you get it? 8 protons, 8 neutrons, 8 electrons. And so if we go to the periodic table, here it is, and here's oxygen, and it has an 8 right here. So that atomic number is the number of protons. If the number of protons in an element changes due to decay or something else, then the actual element is changed. And so it becomes something different. And in here, the mass is 15.99, so very close to 16. So if I take the mass, and I know that electrons are negligible, and subtract the number of protons, so 16 minus 8 would give me 8 neutrons. If it's not an ion, if it is a non-charged molecule, then I know that it's going to have 8 electrons, because the number of electrons equals the number of protons. So that's pretty cool. All right, what are some other things I can see from this periodic table? It's in group 16, it's over here, it's a it's a gas, etc. So you can get into that more of it, but we're not going to be worried too much about periodic table trends in biology. So here is a famous model called the Bohr model. And there's been more models since then, and we know that this isn't quite true with s, p, d, and f orbitals, but for our purposes, it works well. And it works well because we can think about atoms and energy and what's going on. So let's look at argon. Go back to your periodic table. Argon's number is 18. So ask yourself, what does that atomic number mean? Hopefully you got 18 protons, and it's going to have 18 electrons. In the first orbital around the nucleus can go 2 electrons. This is called the first s orbital. In the second orbital around the nucleus can go 8 electrons. And so let's see if there's 8 in there. 1, 2, 3, 4, 5, 6, 7, 8. For those of you that have had a little more advanced chemistry, you know that there's actually an s orbital within the second one, and then 3 different p orbitals aligning on the x, y, and z axis, which gives you a total of 8 possibilities. So now we have 10 so far. Well, let's look at this third orbital. And so here we go. If we know its atomic number is 18, we're looking for 18 electrons. Well, there's 10 so far. We have 1, 2, and then we had the 8 on the second one. So that's 10, 11, 12, 13, 14, 15, 16, 17, 18. Argon has 8 valence electrons on its outermost level. And so those 8 valence electrons make it happy. And you might remember from earlier classes that these are the noble gases, that they're hanging out, they're cool. One danger, and Mr. Glenns would call me out on this, is anthropomorphizing atoms and molecules, saying things like they want, or they're hanging out, or they're cool, etc. When we ascribe human attributes to them, we kind of do them a disservice because they're not humans. These are atoms that are going to react in certain ways, and we're trying to understand them. And there's a lot of things we don't understand. Like if you go back to this and just pose the question, well, isn't there empty space here? The answer is yes. It's so crazy, right? And so all the protons and neutrons are here, and electrons are out here, and then they still interact because the electrons have a charge. And so there's some really deep questions involved in chemistry and physics, and deep questions in biology as well. All right, so let's look at some of these. And here's some of the elements that I've highlighted from Khan Academy. And so hydrogen and helium. So helium will be stable because it fills this shell. So it needed 2 there. Lithium has 1 extra electron in this valence electron shell. So lithium is going to be quite reactive, whereas neon has 8. So neon's good. Fluorine has 7. So if it has 7 and it wants 8, it's going to want 1 more. All right. Models evolve as technology develops. So this is just showing you that, we can move on from the Bohr model, and we can understand that really instead of like orbitals like going around the sun, that there's orbitals in different directions and they have different shapes. And so they can be circular shaped, or it can be shaped like dumbbells, or even other shapes. They come up. So sticking with the Bohr model, where you have 2 in the first level, 8 in the second level, and up to 18 in the third level, can you draw the Bohr models for the elements of life? Sponge. And I've ripped out their periodic table information here. So go ahead and pause the video and see what you can get. All right, let's go through it. So here you go for hydrogen. We've already done that once. It's got 1 proton and 1 electron. Carbon. Carbon has 2 on the first level and then 4 on the outer level. Carbon's like your friend that makes friends with everybody, right? And so it can bond with lots of other elements in order to fill it up and get these 8 valence electrons. Let's look at nitrogen. Nitrogen has 2 here, so it's got 7 electrons, right? And so it's going to have 5 on its outer level. So it's missing 3 to get to 8. Let's look at oxygen. 2 on the first level, and then 6 on the next level. So it's missing 2. Let's look at phosphorus. Phosphorus has 15. So 2, and then 8 on the next level. So that's 10, and then 5 out here. So it's missing 3. So kind of similar to nitrogen. And then let's look at sulfur. It's just got 1 more than phosphorus. So it's got 2 on the inside, 8, so that's 10, and then 6 on the outside. Okay, those are Bohr models, and they're going to be good models for showing us how atoms bond. and how they react together, which is interesting for us in biology. But real quick, you might have noticed something. Why are these numbers with these decimals here? And that begs the question of what is an isotope? And I put Homer in here for you. Okay, so did you get that an isotope is an atom that has a different number of neutrons, right? And so the atomic number is the number of protons, which makes it an element, but they may have different numbers of neutrons. And then their relative abundance on Earth, if you add them together, is what makes the atomic mass. So you would say, okay, let's look at the atomic mass of carbon. It's very close to 12, but not quite 12. So if I know that there's two isotopes, carbon 12 and carbon 13, there's going to be a lot more of this in the world than there is carbon 13 because the atomic mass is a lot closer to 12. You have to think about how you make an average there. All right, and here are the isotopes of carbon. And it's really interesting because we can use it for carbon dating, which I'm sure you've heard of before with fossils. And so carbon 12 and carbon 13 and carbon 14 all exist in the atmosphere, and you have some of these in you right now, all of these different isotopes. And so when carbon 14 is unstable and carbon 12 and 13 are stable. So carbon 12 and 13 can just exist as carbon 12 and 13 forever. But carbon 14 is unstable, and so it will start to decay. It will undergo radioactive decay, and it will turn into nitrogen. What does that mean? It means that it's going to undergo beta decay, and one neutron is going to change into a proton, going from having 6 protons to 7 protons, which makes it turn into nitrogen. Pretty crazy, huh? This is pretty interesting because you can look at, say, a bone or a fossil, and you can look at it and then say, okay, it has this amount of carbon 14 in it, or it has this amount of nitrogen 14 in it, and you can figure out how old it is. Because once the organism dies, it will stop eating plants or eating other animals that ate plants. And so it'll stop intaking carbon 14. So from that moment onwards, that carbon 14 inside of it will start to decay. We'll get into that more later on. Don't worry about that. I don't know if I said that the most elegantly. All right, so I've been hinting at these rules and these outer shells. So the outer shell is called the valence shell, the ones that interact. And the octet rule means that 8 is great, is what I used to tell my students when I taught like a beginner physical science class. And so it means that the atoms are stable when they have 8 in their outer shell. So we've gone over that. So here you can see that neon is very stable. 1, 2, 3, 4, 5, 6, 7, 8. Whereas fluorine is missing 1. So fluorine is going, and here's where I'm running that risk of making them sound like humans again, but it's going to try and get 1 more electron. So let's check some Khan Academy, and I got this from their website because I think it's very good. So hydrogen, lithium, and sodium are group 1 elements, and they have 1 electron in their outermost shells. So they're unstable, and they're more likely to give these up. Here they are, hydrogen, lithium, and sodium. So you can see where I got them from. They just have 1 in that outer shell. If they give it up, they're going to become what's called a cation. And a cation, I just think about it as a cat that's purring. It is positive. So here, the sodium gave up that electron. But what does it give it up to? It has to give it up to something. You can't just give it up forever. And so here is a good example. Fluorine and chlorine are group 17 elements. They have 7 electrons in their valence shell, and they want 1 more. So let's look at the picture. Here is fluorine and chlorine. And so they want 1 more, so they could get it from sodium or lithium. And so here, fluorine is going to gain that extra electron. It's now going to have 8 in its outer shell, and it's going to be negatively charged, and it's called an anion. Well, opposites attract, and this cation and this anion right here of sodium and fluorine would combine together, and they would make what's called an ionic bond, and that could be sodium fluoride. You've probably seen one with sodium and chlorine, and that would be salt, NaCl. Carbon is a group 14 element, it has 4 in its outer shell, and like I told you earlier, to achieve a complete valence shell, it's going to form bonds with multiple other atoms, and that's why carbon is the backbone of so many different molecules. Atoms interact with other atoms to form bonds. Okay, so here's where it gets interesting. And here's where, biologists can be naughty. And naughty and I've talked to Mr. Kelly about this, we tend to say things like breaking bonds releases energy. And it's all tricky. And it's tricky because atoms are constantly trying to get to their most stable or lowest energy state, their full valence shells. So it's actually the formation of very strong bonds that releases energy. We'll get into that later on. Don't let it confuse you too much. You can still think of ATP breaking that bond as releasing energy, and we'll go over it more later on. All right, so here's some types of bonds you may have heard of before. Let me move my head and my hair. Ionic, covalent, hydrogen, van der Waals, London dispersion sources, and hydrophobic interactions. So I've kind of grouped them here, and hopefully we've heard that ionic and covalent bonds are strong, and that hydrogen, van der Waals, and hydrophobic interactions are more weak. All right, but they're all really important. You wouldn't want some molecules bonded together covalently in your body if you need to be able to break them apart and easily access them. And some of them you do want bonded together covalently or ionically. So all of these work together to make life as we know it. So let's do some examples of an ionic bond. Here is the one on salt that I told you about earlier. So salt's going to give up that one, sodium is going to give up the one valence electron in its outer shell. Chlorine will grab it. It makes sodium a cation. It makes chlorine an anion. And they have a bond because they are happy as opposites attract. Here's an example of an ionic bond within a protein. So proteins are made up of amino acids. The backbone of an amino acid is the same, but the R group or the side chain is different for each one. So here is an R group that's going to end in NH3+, and here's one that's going to end in O negative. So can you see this would be the cation, here would be the anion, so then there'd be a bond or an ionic bond forming between those opposite charges. And that can help give our protein shape, and the shape of a protein leads to its function, which is really important. Okay. Covalent bonds, I just remember the C in front of covalent for being like a couple, and a couple should share. Here's an example of oxygen and hydrogen, and this is really important and part of your summer assignment, right? So oxygen and hydrogen, and once again, I'm doing the risk of making them sound like people, but hydrogen had one valence electron, so one and one more. Then it would have two in that shell. Here, this hydrogen, the same thing. Oxygen wanted two more electrons. If you remember, it had a valence electron of 6. Here, I thought I had it right there. I'm sorry. Well, it has a valence electron of 6. So it wants two more. And so here, by sharing with these hydrogens, it's able to have the correct number. And so this is pretty interesting. Look at it. If you look at oxygen, it has 8. And if you look at hydrogen, it has 2. Remember that the first orbital only needs 2. So now here's an interesting question. Do they share evenly? By that I mean, is this polar or nonpolar? Do you have any idea? Well, the way you can think about it is just polar means it has poles. It has positive and negative ends. And so oxygen is a larger element. It has 8 protons on the inside, whereas hydrogen only has 1. And so the electrons will be around the oxygen more often because oxygen has a bigger pool for it, a bigger number of protons, so it's stronger. It is what we call the more electronegative element. It's going to attract those electrons. So therefore, in a water molecule, the hydrogens will tend to be more positive and the oxygen will tend to be more negative, which makes it polar. Here's an example of a nonpolar molecule, methane. So methane is CH4. It's going to have 4 hydrogens coming off of it. It's an equal sharing of electrons. It is nonpolar. It does not have poles. Here's an interesting one, carbon dioxide, right? And so carbon dioxide, causer of the greenhouse effect, etc. It's going to be nonpolar because it's going to equally share these electrons and it's not going to have poles. So how can you figure this out? You will not need to do this in AP biology, but you'll do it in chemistry. You can look at electronegativity values. You'll see that the most electronegative element on the periodic table is fluorine. So there's many memes about that, but oxygen's right near it, so it's pretty close. Electronegative means it's going to pull electrons towards it. If you look at elements and then you look at the difference between them, you can figure out what type of bond it's going to form. Here, the difference between oxygen and hydrogen, 3.5, and hydrogen being 2.1 would be 1.4. So it's going to become a polar covalent bond. And that means that it's going to be polar. Whereas if it's two oxygens bonding together, because oxygen exists as a diatomic, then it's going to be less than that. It's going to be covalent, so it's going to be nonpolar. So oxygen doesn't exist just on its own, right? It would need two more valence electrons, so oxygen can bond to itself to make O2. Another example of that would be hydrogen. Hydrogen doesn't just exist on its own. It would bond with itself to make H2. So that would be 2.1 minus 2.1, it'd be 0. Now here's where it gets interesting. There's all kinds of exceptions. Right? If you look at CO2, look at the difference between carbon and oxygen. Carbon 2.5, oxygen 3.5, it's a difference of 1. You think it should be polar, but it's not. And it's not polar because it forms a linear molecule where it's able to distribute that charge evenly. So there's some differences in there, and there's some stuff that gets beyond the chemistry involved in this course. So usually the AP biology test will tell you whether something is polar or nonpolar, which is important because things that are nonpolar may diffuse through the cellular membrane, and things that are polar cannot. Okay, so that was covalent, and then covalent bonds can either be polar or nonpolar. We said ionic bonds are between anions and cations. So now we have to learn about those weaker bonds. And one of them that's going to be really important are hydrogen bonds. And hopefully you recognize this. You see the T's, the A's, the G's, and the C's, and you recognize this as DNA. Here's the sugar phosphate backbone, and then here's the insides. These sugar phosphate backbones are held together by covalent bonds. Covalent bonds are strong bonds, as we said earlier. But the bases, or the nitrogenous bases that code for proteins, that give us who we are, you are the summation of all the proteins that come out of that are coded for by your DNA, are held together by weaker hydrogen bonds. A hydrogen bond, just like you can see in this picture here, is going to be the more positive hydrogen is going to be attracted to a more negative element. And the negative elements that we will think about are oxygen, nitrogen, and fluorine. So let's look right here. This more positive hydrogen is going to be attracted to this more negative nitrogen. This more positive hydrogen is going to be attracted to this more negative oxygen. Here, this hydrogen attracted to this nitrogen, this hydrogen attracted to this oxygen. That is called hydrogen bonding. You can see it between water molecules as well, which is why if you hold your finger up with some water on it, the water sticks to itself or sticks to a surface, because the positive H's in the water molecules will temporarily bond with the negative O's in another water molecule. Now those bonds can be broken, and they are broken all the time, but they're strong enough to hold together the water molecule and to give it some type of cohesion or to give it surface tension, which is really interesting. It's really important too for how plants work and how transpiration works to get water up a plant. Okay, last but not least, there will be chemical reactions, and we'll go over these a lot, especially in Unit 3 with cellular energetics. And here's a very famous one that many of you have done an experiment with before, and if you haven't, we will do a fun one in class. So hydrogen peroxide becoming H2O, or water and oxygen. So hydrogen peroxide is a waste product in your cells. It's also, you can buy it here in a pharmacy, and it used as a disinfectant. And with the help of an enzyme called catalase, it can be broken down into water and oxygen. So you're going to have coefficients and subscripts. You're going to need to be able to add up those atoms and make sure that they're equal on each side. And this is why when you walk into a restaurant and ask for water, somebody says H2O, and then the next person says, give me a H2O22, well, then you know you're in trouble, right? Because you're going to get hydrogen peroxide. Dad joke there. This is pretty important because it deals, sometimes the arrows will go both ways. And so here's an example of a buffer. And so this is in your blood. And as you'll read, and work on later on with Unit 1, you'll see that hydrogen protons, these H pluses, make something acidic. And so the more of them there are, the more acidic something is. The less of them that are there, the more basic or alkaline something is. So if you add too much, or if you get a whole bunch of H pluses in the system, then they can combine with bicarbonate ion buffer, this, to become this, carbonic acid. And then that carbonic acid can, in your lungs, be getting rid of in the form of CO2. So it helps to modulate where your blood pH is. So that is really important because your blood is full of many proteins that carry oxygen. And so if those proteins got denatured in the wrong pH, they wouldn't work as well. All right, and this goes the other way as well. If it becomes too basic, then this can dissociate into carbonic acid and more hydrogen ions, therefore making it more acidic. All right, where are some examples of all this stuff in action? This is from OpenStack's proteins. Proteins are a huge portion of AP biology. So here, the interactions between different amino acids on the protein. Here we can see a covalent bond between two amino acids that have sulfur in them, which are either methionine or cysteine. You can see hydrophobic interactions. That means they're trying to get away from water. So they go towards the inside of the protein. Here you can see a hydrogen bond between the H and the O of these different amino acids. And here's an ionic bond, as we showed earlier. All right, students, that was my pre-AP Biology Chemistry review. Reach out to me over the summer. I am available for help. And good luck.